Hello, friends. Let's start with this topic on physiology of human digestion. Since the topic is physiology of human digestion, it means that diagrams of the alimentary canal is not expected. Diagrams of the alimentary canal is not expected. So, as we know, we will look at it like this. Food of human beings is comprised of three parts: carbohydrates, proteins, and fats. These are the three main components, and we will look at this topic from this point of view, that is, how the carbs are digested, how the proteins are digested, and how the fats are digested. So I think that it will be better if you can find out first what are the components of carbohydrates, what are the components of proteins, and what are the components of fats. In very brief, carbohydrates means naturally they contain a lot of they are nothing but hydrates of carbon, so they will contain monosaccharides such as glucose, fructose, galactose, etc. These are made into polymeric compounds like starch or glycogen or cellulose, whereas proteins, they are made up of amino acids, and fats are made up of fatty acids and glycerol. So let's start with this topic. The first component of the human digestive system is that it contains mouth. Mouth contains teeth, which is used for biting, crushing the food. Then there is glands are present, which we call as salivary glands. So the salivary glands contain or they are made up of parotid glands, sublingual glands, mandibular glands. Most of the time salivary glands release enzyme called salivary amylase, which works under alkaline conditions. Means the optimum pH is likely to be over 7. pH of around 7.6 is the optimum pH. And salivary amylase digests carbohydrates. So if we have polysaccharides, then polysaccharides in the presence of salivary amylase will convert them into, or will try to convert them into, maltose, which is a disaccharide. Not all of it happens, some of it happens. We have to therefore give time for the polysaccharides to get digested into disaccharides. So that's why chewing of food is so important. Chew food properly. Slowly, and therefore this digestion can take place. Okay, this is about the mouth. from here the food passes down into esophagus, but in the esophagus no digestion. The food only goes down by peristalsis. Peristalsis will also work even if the person is standing upside down. So it is regardless of the person's position, whether that person's head is on the top or head is below, it will still work. So this is peristalsis. Naturally, due to some problem, if that person has to vomit the food, then that is called antiperistalsis. So this is waves of contraction and relaxation will push the food down towards the stomach. This is because of the muscles, action of the smooth muscles in the esophagus and in the rest of the alimentary canal, wherever it has to be pushed forward. Okay. The third component is stomach. So from the esophagus, the food moves into the stomach. Stomach is a bag which is highly extendable. That means when nothing is eaten, it is quite small. But when we go on eating, it can become bigger and bigger, substantially bigger. And the stomach is folded into large folds are there. These folds are called as rugae. Large folds are there called rugae. Stomach is also very muscular. Different sets of muscles are present which can churn the food. Digestion, so the food in the stomach remains for about 3 to 4 hours inside the stomach. Now, these rugae which are there, these rugae are further folded, and in the cellular layers we get like these kinds of, these are called gastric pits. These contain these glands. So they contain gastric glands. And there are many cells lining this part and cells lining this area. So cells lining here, some of these cells over here, these are called as chief cells. And the cells on the sides, they are called as parietal cells. Parietal means side. I am not drawing the diagram because it's not expected. So these chief cells will release pepsinogen, and parietal cells will release HCl. Now we will come across this term such as, say, pepsinogen, and later on trypsinogen, chymotrypsinogen. These are all called as zymogen granules. That means these are the inactive or precursor molecules of enzymes. They will become active only when in the presence of some conditions. So in this case, pepsinogen is converted to pepsin in the presence of HCl when there is food. this HCl very acidic. This makes the pH of the stomach acidic. This you should remember. Okay. The pH is likely to be around a value of 2 or less than 2, or an average around 2. So this is very acidic. It's the roughly any cleaning solution in the house that you are using, that pH is 2. So the immediate question is, if this is so acidic, why does it not destroy the cells of the stomach? The answer is that these gastric glands, they also produce a lot of mucus, and this mucus has a protective function. The mucus has a protective function to protect it from its own acidity. And the important thing is that acid is released only when there is food, or there is a time for the food to be had. Naturally, this timing is very important, and if, as nowadays, we don't restrict ourselves to specific timings, then this acidity from the stomach moves up through the esophagus, and we get problems of acidity and hyperacidity. So these are disorders of the stomach, especially of acidity. So pepsinogen in the presence of HCl is converted to pepsin, its active form, and then all the proteins are converted by pepsin into peptides. So you see, let's just suppose we draw a protein of a particular shape, something like this, and the pepsin will cut it up into at different points. Something like that. And this pepsin is all these enzymes cut at specific points. So pepsin is a type of protease because it cuts the protein, therefore the word is protease. So see, last three letters are ASE. It cuts the protein in between, therefore it is a protease. You can also say that it is an endoprotease. It's just the name that was given, early name, therefore that name remains. But if you want to use a more specific term, you can call pepsin as a protease or an endoprotease because it is cutting internally. If it cuts externally, we use the word exoprotease. So this is the only digestion that takes place in the stomach. The rest of the food, because of the churning action of the stomach, it is still broken down into small and small pieces. And it remains for a couple of hours, and then the food then passes into the next part, which is intestine. It will pass through into the intestine through a sphincter. Okay, sphincter is like a ring of circular muscle which guards it. So it moves from here to here. And this intestine now has got one part which we call as duodenum, then a part called jejunum, and then a part called ileum. Note the spelling of this, Ileum. If you spell it differently, it will mean bone. Okay, the iliac bone. But we are not talking about the iliac bone, it is Ileum. So there are three parts: duodenum, jejunum, and ileum. But generally, we don't regard this. We will put this in, we will understand that the jejunum is part of the ileum and just say two parts are there, essentially, just for understanding purposes. So, duodenum and the ileum, both of them together is alkaline. So from the stomach which was acidic, it has become alkaline. That only then will the food gets digested. So this change of condition is important. There under acidic conditions it will work. All the enzymes over here work under alkaline conditions. Now duodenum, this will receive pancreatic juice through the pancreatic duct, and this pancreas releases, or it has three main enzymes. One is chymotrypsinogen, carboxypeptidase. Chymotrypsinogen, trypsinogen, and carboxypeptidase. All these are protein digesting enzymes. Now in the duodenum, so in the lining of the intestine, we are talking about the intestine now. So in the intestine, there is an enzyme called enterokinase, which is a membrane enzyme, membrane of the intestine. So this converts trypsinogen into trypsin. This becomes active. Now trypsin converts chymotrypsinogen into chymotrypsin. And this also, which was in the inactive form, becomes active in the presence of this. So this becomes carboxypeptidase. So let's see what we mean by carboxypeptidase, why it is called like that. So you see, if there is a protein, we show different amino acids like this. Protein will always begin with amino terminal with N and will end with C terminal, carboxy terminal, NH2 and COH. So amino terminal and carboxy terminal, and these are shown as amino acids. So when we take an enzyme like carboxypeptidase, what it means is that the enzyme will come and attack it first over here, then over here, then over here, then over here, then over here. That means carboxypeptidase will release amino acids successively from this end. Similarly, in the intestine, that is in the ileum, there is also aminopeptidase. Aminopeptidase will release amino acids from this end. This will also give rise to amino acids. And if there is a dipeptidase, means there are two amino acids, it will break it down and give rise to amino acids. There are two amino acids joined together, that's why the word is dipeptide. If there is a dipeptide bond, it will break it and give it to two amino acids. So whenever we use the word peptidase, it means that it is going to give rise to amino acids. Whereas enzymes like chymotrypsinogen or trypsinogen, which will be converted into trypsin and chymotrypsin, they will give rise to peptides, short peptides. Okay. So due to the action of enzymes like trypsin, so proteins they are acted upon by trypsin and chymotrypsin to give rise to different short peptides. You can use the word oligopeptides over here. But you will ask why this? Because this has a different point of action between two different amino acids. This has a different point of action between two different amino acids. For example, this works between, say, glycine and lysine. This works between, say, phenylalanine and lysine. So the point of action is different. It will cut it up into different lengths. Peptides of different lengths. So these peptides now will be reacted upon by carboxypeptidase and will give rise to some amino acids. Similarly, the same thing by aminopeptidase. But aminopeptidase comes from intestine, carboxypeptidase comes from pancreas. That's the only difference. Again, into amino acids. Then there may be some dipeptides. These also are acted upon by dipeptidase. And these are giving rise to amino acids. So in this way, by the time we come to intestine, that is the ileum, amino acids have been formed, and amino acids are then absorbed by blood vessels, blood capillaries. Where are these blood capillaries? See, the intestine is a long tube, very small diameter. Barely about one inch or so, and it is folded into villi, and in the cellular section these villi are subfolded into microvilli. It is nothing but the brush border of cells. So on one side we have microvilli and on the other side we have blood capillaries. So the amino acids are easily transported from microvilli into blood capillaries. Okay. Remember that we are still talking about intestine, and we are talked now only about proteins. What happens to the fats? The fats have not been much digested in the stomach, in fact no digestion at all. In the mouth there is a little bit of salivary lipase which digests it, but majority of it comes down into the intestine. When it comes to the intestine, duodenum also receives bile duct. Also receives bile duct. So this contains bile juice, and it contains salts, bile salts, for example, sodium taurocholate, which will emulsify, not digest. Means break it down into small globules, fats. Fats are also called lipids. So fats, bile, salts, they are emulsified fats, means broken down into smaller globules. Then these emulsified fats, they are converted by lipase into fatty acids plus glycerol. And this lipase is intestinal lipase. So in the intestine further digestion takes place of the fats. So fats are converted into fatty acids and glycerol. But this is not soluble in water, that is this fatty acid, and therefore it is absorbed and passed across through lacteals. So these are lymph vessels or lymph capillaries. Into blood vessels. It is from the lacteals across the portal system, across the intestine into the portal system. This is a different type of a blood vessel which we call as a lymph capillary. Now remember that we said that in the mouth carbohydrates are digested into maltose units or disaccharides. So in the pancreas now there is a little bit of pancreatic amylase, or you can simply call this as intestinal amylase, which will also further digest carbohydrates. Some amount of the polysaccharides were still undigested, and these get digested into disaccharides. So in the intestine further there are enzymes like maltase, ASC enzyme. This will help convert maltose into glucose, monosaccharide. Okay. If there is substrate called sucrose, then it is digested by sucrase into glucose and fructose. Similarly, there is milk, sugar, lactose, enzyme is lactase, glucose and galactose. So in this way, all these are from the intestinal enzymes that digest polysaccharides and they convert them into monosaccharides. So these are all absorbed by blood. Same way, just as proteins were absorbed by blood across microvilli, then into the portal system. So all the products, okay, what are the products of protein digestion? Amino acids. What are the products of fat digestion? Fatty acids and glycerol. What are the products of carbohydrate digestion? Monosaccharides. So all these are taken into to liver by or across portal system. Since it is liver, it is hepatic portal system. Then it is detoxified over there. So majority of the digestion and absorption takes place inside the intestine. So we have learnt this particular part, that is digestion in stomach and digestion in intestine. In this way, you can also do it in another way, digestion of proteins, digestion of carbs, digestion of fats. What I suggest you do is, say, take protein and write from mouth up to intestine what digestion takes place. Write about that, a note about that. Similarly, carbohydrates from mouth to intestine, how it digests, from fats, how it digests. So learn it in this way also, learn it from the organ point also. That will be helping you to convert or understand this topic. So with that, I'd like to end this particular lecture, and this is Professor Ravindranath signing off. Thank you.